Today we have a presentation given by two experts in the global COVID-19 pandemic. The seminar today is COVID-19, a global perspective. So the first speaker is Professor Saverio Stranges from the Western University, London, Ontario, in Canada. Professor Saverio is chair of the Department of Epidemiology and Biostatistics at the Schulich School of Medicine and Dentistry. He's a medical doctor, a public health specialist and chronic disease epidemiologist with extensive experience in the field of epidemiology and public health research. He completed his medical education in 1996 at the University of Naples, Federico II, with a specialty training in preventive public health medicine. He started his academic career as assistant professor at the State University of New York in Buffalo in 2004, and then moved to the University of Warwick and Warwick Medical School in the United Kingdom from 2006 to 2015, where we were colleagues. He was then scientific director of the Department of Public Health at the Luxembourg Institute of Health in Luxembourg, and then moved to his current position. His research focuses on epidemiology and prevention of chronic disease and aging. He's interested in global health, and then, of course, became very interested this year in the development of the COVID-19 pandemic. He has over 180 publications, H-index 58, and over 25,000 citations. If you look at the publications this year, you'll see he's been very active in COVID-19 pandemic research, publishing on excess mortality from COVID-19, a commentary on the Italian experience, where, like many Italianophiles, friends of Italy like myself, he was very disturbed to see the high mortality that COVID-19 has brought to northern Italy. And then also an article on COVID-19 pandemic. What can the West learn from the East? Because Eastern countries have done very well, Far East, in managing the COVID-19 pandemic with respect to public health initially. And then also a call to action about SARS-CoV-2 infection and cerebrovascular diseases, COVID-19 and inequality, social inequality, and the ethics of COVID-19 related school closures. So he's a world expert in public health and now is very much fascinated and interested in how we're handling the COVID-19 pandemic. We're also fortunate today to have a colleague of Professor Saverio's, Professor Eric Arts from the Department of Microbiology and Immunology. Professor Arts is an expert in viral infections and has done much research in HIV infection. And he will talk about drugs and vaccine development research. Professor Eric did his BSc first at Western University before going to McGill for a PhD and then to Case Western Reserve, where he started HIV research. So he's come home to his former university alma mater to pursue his research in virology and immunology. So welcome both, and we're very much looking forward to your presentations. Professor Saverio first, and then Professor Eric will take over.
Join you today, and so I'm pleased by this invitation. Paul, it's nice to reconnect with you after our time in Warwick. So I will, let me share my slides. Hopefully it will work. Seems that it's working now. Okay, so yes, I will provide an overview, global perspective on the current COVID-19 pandemic. And my friend and colleague Eric Arts, who has more expertise than me in virology, microbiology, will discuss about specific issues around vaccine developments and drug options in this particular field. And let me see if I can move the slides. So first, I just wanted to provide an overview on Western University, because Many people may not know there is a London also across the ocean, which is in southwest Ontario, and you see London lies in between New York State and Michigan State in US. Is one of the most southern places in Canada. In fact, we enjoy relatively mild temperature, and Western University is one of the top public university in Canada, a long-standing tradition in the medical school. We have a large number of students and also over 4,000 full-time staff. It's a very green city with parks surrounding the city as well. So this is the outline of my our presentation today. As I mentioned, I will discuss about global epidemiological trends, public health responses from an international perspective, and then I will leave to Eric to talk about vaccine developments, pharmacological options, and testing. And we want to make sure to leave enough time for questions and open discussion. Obviously, COVID-19 is only one of the last viruses within the coronaviruses family. And if you can see the timeline coronaviruses related publications in PubMed, you can see that there has been quite a growth in the number of publications starting from the 80s onward. So the emergence of coronaviruses has been a major public health issue over the last 20 years or so. And obviously SARS-CoV-2 is only the latest of these viral infections. Is likely the greatest public health crisis since 1918 influenza pandemic, the so-called Spanish flu. The case fatality rates tend to vary, but seems to be currently around just over 3% globally, although I will discuss about the limitation of using this measure when in the current pandemic. But as I mentioned before, actually the globe was already affected by other coronavirus infections like MERS-CoV, which happened over the last few years, called also the Middle Eastern Respiratory Syndrome coronavirus with over 2,000 cases, 2,500. But the main difference as compared to SARS-CoV-2 was the very high fatality rates, nearly 35%. And there was very little impact, as you can see in North America, including US and Canada, sporadic cases in Saudi Arabia. But back to over 20 years ago, nearly 20 years ago, we also had SARS-CoV, the very first CoV infection, which actually affected over 8,000 people worldwide. Again, the case fatality rate was much higher than the current COVID-19 infection, being at 9.5%. In Canada, there were also a few hundred cases, but that infection, again, affected by very high case fatality rate, was primarily hospital-based infection. So there was very little community. So what is specific and why is so hard this current pandemic with COVID-19? Well, we're talking about highly infectious virus for about 7-10 days, tends to be infectious even before the onset of symptoms, which is relatively uncommon for respiratory virus. And again, Eric will discuss these aspects. The initial presentation also wide-ranging and non-specific, which make obviously difficult to diagnose in a timely fashion at times, and also easy to mistake for something minor. 15-30% of patients remain asymptomatic throughout infection, but they have the potential to infect other people for at least days. But can also be severe because we know that a certain number of patients, up to 5%, may require mechanical ventilation, and 0.5% of patients require dialysis. Obviously, these figures are driven also by the specific demographic structure of the population affected. But we need to put things in context. COVID-19 has been a major public health crisis. There is still sometimes comments about relatively little impact of COVID-19. But if you look at, for example, at the average total death in US between February and May this year as compared to historical figures from 2014 and 2018, you will see that COVID-19 actually stands as the third cause of mortality within the US context, just behind heart disease and cancer. So despite the relatively low case fatality rate, COVID-19 has caused at least 5 times higher number of deaths than, for example, the common influenza pneumonia. So where we stand in terms of the current epidemiological scenario globally, and then we go also into specific data from Qatar and Canada. You can see that the number of cases is increasing from January to date, and I think just 2 days ago there was the largest number of cases recorded, over 300,000. So the pandemic is still happening, and we are still experiencing the peak of the pandemic. In terms of the number of deaths, which currently stands at 927, based on September 13th data, well, you can see that there was a peak in terms of the number of deaths happening around April, May time, where the pandemic was affecting especially European countries, which are characterized by much older populations. And then as the pandemic spread also in low middle income countries, you see that the number of daily deaths has been relatively stable, definitely lower than in the first wave, affecting, as I say, European countries and also US, North American countries in the first place, because again, obviously the population demographics tend to change, to vary across the country. So if we look at, this is a report which was published by WHO just yesterday. So I updated the figures last night. You can see the number of COVID-19 cases reported weekly, and this is from December through 13 September 2020. And you can see that how the pandemic has been affecting different regions of the world, switching from high-income countries, including obviously North America and Europe and Western Pacific as well, and now becoming a major crisis, especially in Southeast Asia, countries like India in particular being affected. Also, African countries are now experiencing cases of COVID, although the toll in terms of death is still quite limited, possibly because of demographic of the population as well. And so the current scenario, at least for the last week, in terms of distribution of cases of COVID-19 per million populations globally, and you can see how from the dark spots that South America is currently possibly the epicenter of the pandemic. But as I mentioned before, the pandemic is affecting also African countries, and as you know, in Europe and now possibly also in North America, there is a surge again of cases as we reopen the economy over the last few months. So I was also asked about talking on the Qatar situation, and I was impressed by a couple of distinctive features of Qatar. First of all, Qatar is one of the countries with the largest or highest rates of testing per million population, standing at nearly 250,000 tests per million population, which is remarkable. And the number of cases, as you can see, just over 120,000. And it seems that if you look at the daily cases, it seems that at the moment the situation is relatively stable, with relatively few cases on a daily basis. What is also very interesting to notice is that number of deaths in Qatar has been relatively small, 205 as of September 13th. And if you look at the number of deaths per million population, which is a much solid epidemiological indicator to make comparisons also across countries, you will see that Qatar is a relatively small death toll as compared, for example, to Western countries, including Canada. In fact, when we look at Canada, for example, we're in a country of now nearly 38 million, Qatar is 2.8. We have just a slightly higher number of cases, but you see that the testing in Canada has been less than in Qatar, just over 160,000, and especially in the first few months, we were struggling in terms of testing capacity, and I will discuss about these. And when we look at the number of total deaths in Canada, at the moment we had over 9,000 deaths. And again, if you look at the number of deaths per million population, we have 243 deaths, which is more than 3 times higher than Qatar. And Canada is considered a Western country where actually the response to pandemic has been relatively successful in mitigating the number of deaths. If we consider, for example, US, also other Western European countries in Spain, Italy, or England, the death per million population is nearly close to 600 per million population, if not higher. One particular vulnerability of the Canadian response to COVID-19 has been the relatively high number of deaths in long-term care facilities. Over 80% of all deaths in Canada actually happen in long-term care facilities. Now, this has been a common challenge for many Western countries where the policy responses within a long-term care facility has been relatively poor. For example, in Canada, France, Norway, Spain, US, and UK, with a relatively limited attention also in terms of, for example, availability of PPE, especially at the very early stage of the pandemic. On the other end of the spectrum, you will see countries like Austria, Australia, or Slovenia, where actually the policy response has been the highest possible within long-term care facility, for example, with isolation wards, with a broad testing, also with infection control training audit, including the presence of qualified personnel, and therefore there has been a rapid response control and prevention teams which have been able to really mitigate impacts of COVID-19 within these settings. So as Paul was mentioning a few months ago with some of our former students from our department, we wrote an article, Perspectives on COVID-19 Pandemic, What Can the West Learn from the East? And this was at the time, back in June, where obviously there was a clear gradient in terms of impact of COVID between the West and the East of the globe. So we said the differences in public health approaches to control COVID-19 pandemic could largely explain these substantial variations in epidemiological indicators such as incidence and mortality between the West and East. The pandemic revealed vulnerabilities of most Western countries' healthcare systems in their response to the ongoing public health crisis. For example, limit testing capacity at first. Specifically, there was also lack of rapid and timely community-centered approaches, and most importantly, weak public health infrastructures might have resulted in a high number of infected cases and fatalities in many Western countries. Hence, Western countries could possibly learn from practices from several East Asian countries regarding infrastructures, epidemiological surveillance, and control strategies to mitigate the public health impact of the pandemic. One issue that Western countries face in terms of the COVID-19 pandemic has been the issue of flattening the curve in order to avoid overwhelming healthcare systems, especially hospital settings. We always say that pandemic and infectious disease outbreaks or epidemics should be handled in the first place within community settings if there is a strong public health infrastructure which will do proper surveillance and isolation of cases. If that does not happen, that's where you have the pressure on hospital settings, and that can actually overwhelm the capacity of the system to cope with concentrated number of cases of new viral infections. And so this also links to the issue of critical care beds per capita for selected countries, because despite the fact that in some Western countries like US and Germany had enough number of beds, we're talking here about intensive care units, other countries like Italy, France, Spain, United Kingdom, were with relatively limited capacity in terms of ICU beds to respond to such an acute, overwhelming number of infections, especially over the spring months, from March until May. So some of these Western countries, in a sense, were forced to implement even national lockdowns, and this is my home country. Obviously, it's been upsetting to see the images of historical places in Italy completely empty during the lockdown situation. So we tried also to explain the reasons for the excess mortality from COVID-19 within the Italian context. And I'm also citing here a paper from one colleague from the University of Bologna about some of the intrinsic issues within the the Italian response in terms of lack of pandemic preparedness, the decentralization of the public health system, which has been an issue in the response to the current pandemic, and also the hunt for patient zero, because at some point it was obvious that actually we missed the boat in terms of controlling community spread in Italy. And so with a surge of cases back in the end of February, actually we struggled to really find the patient zero, also because, and then we learned that most likely the community spread in Western Europe was already happening, at least from the end of January onwards. And in fact, the first European cases actually were detected in Munich, official cases were detected in Munich, Germany in January 2020, with a clear epidemiological link with Chinese business people coming from Shanghai to Munich, Germany. In February, when the possibly community spread was already very high, it was extremely difficult to make epidemiological links to find the so-called patient zero. So, for example, what happened in Italy, and this is relatively similar to other Western European countries, such as England, France, Spain, Belgium, and so on, there has been a very high COVID-19 death toll, which has been driven by the demographic of diseased individuals. You see that in Italy, the median age of diseased people from COVID-19 was over 80 years of age, against the median age of diagnosed cases, which was 64.0. And as Paul was alluding, actually the dramatic increase in the number of death and case fatality rate was primarily driven by the Lombardy region in the north, which is also the region possibly with the highest degree of trade and economic links or trades connections with the rest of the world, including China and other European countries. And in the rest of Italy, actually, the case fatality rate was nearly half of what we experienced in Lombardy region. And again, if you look at distribution of diseased individuals in Italy, you will see that the increase, the number of deaths happened above the age of 70. So how can we explain variations in death toll across countries? Obviously, demographic profile of diseased individuals, much older in Italy, Western European countries than other countries. In particular, Italy has the oldest population in Europe, is second to Japan. On Earth, we have nearly 23% of Italians are 65 years or older, with a also sizable number of people above the age of 80. Lombardy healthcare system has been overwhelmed with a much higher case fatality rate than the rest of Italy. We also need to notice from an epidemiological perspective that there is a discrepancy among countries in distinguishing death from other causes while being infected with COVID, because at the present time, there are no international guidelines around COVID-19 death certification. And also there is a huge degree of cases under detection across countries, depending on testing capacity and approach. And So what we see usually is the tip of the iceberg, which is a basic concept for infectious diseases, where, you know, in most cases we may lose a large number of asymptomatic individuals because what we see are only people with either mild illness or people with severe disease or even death. And so there has been a lot of discussion of what is the amount of under-detection with the COVID-19, and very likely we missed, you know, a large number of cases, especially at the very early stage, where the testing capacity was extremely limited, especially in Western countries. And this lack of detection of asymptomatic cases definitely has been inflating the case fatality rates in Italy and also other Western countries. So these are just the last two slides of my presentation. So lesson learned, a way forward. Physical distancing face masks work, but the applicability and long-term sustainability of these measures in Western countries is uncertain. There has been inconsistent approaches across country for case identification, containment, resource allocation to slow down community spread. We learn from Asian countries like South Korea or Taiwan the importance of a centralized public health infrastructure with proactive testing. It's also important to protect healthcare workers. For example, in Italy, we had a very large number of physicians and nurses who were infected, and a large number actually of health professionals died. At the very early stage of the pandemic, there was also a problem of supply chain, because many Western countries at that time did not produce masks, for example. So there was a shortage of swabs and face masks, which was, as I said, a problem at the early stage of the pandemic in Western countries. And obviously the pandemic is also called for the need to strengthen networks of ICU beds across hospitals with the creation of specific COVID hospital sites. In terms of public health recommendations, we know that physical distancing, face mask, eye protection are very important. Neither of them is a perfect measure to mitigate the risk, but the combination of those two measures ensure a substantial reduction in the risk of infection. And this is a systematic review from colleagues from McMaster University published in The Lancet, which has driven much of the public health recommendations currently implemented around the world. So to finish my part of the talk, I think the COVID-19 pandemic has revealed some of the weaknesses of Western countries. We know that healthcare systems in Western countries are primarily designed to prevent and manage chronic non-communicable diseases. There is a need to expand public and private investments in infectious disease control and surveillance, epidemic preparedness, including vaccine development. There is also a need to refocus the epidemiology and public health training on infectious disease and their interplay with the chronic comorbidities. We need to capitalize on new data technology to facilitate proactive case identification, as has been done in some Asian countries. Another important lesson is the communication with the public, which is crucial to offset the misinformation and fake news in the era of social media. And finally, international partnerships and share of data have been lacking, especially within the European context. So I have finished, and I will let Eric talk now.
Please, Professor Eric, when you're ready. You're muted. You're muted, Professor Eric. We can't hear you.
Thank you. Can you guys hear me now?
Yeah, we can hear you now. It's okay, fine.
Sorry about that. Yeah, so thank you, Saverio. And I'm going to start off straight away with a description of the vaccine developments and other developments in terms of treatments. I was going to present a few things on the Chinese epidemic and how we got to where we were, but I think Saverio did a great job. So I'm just going to jump straight in aspects related to more of the biological and understanding our developments thus far in treatments and vaccine developments. So I think it's fair to say that we've made an incredible amount of progress in a short period of time in dealing with this pandemic. And I think all of you read the news or hear the news in regards to all of the developments in moving vaccines to a point of phase 3 clinical trials in a record period of time. I think there are caveats in understanding that aspect, those being that it is remarkably easy in retrospect to develop a vaccine related to SARS-CoV-2 in relation to other viral infections that we see in the pandemic today. In other viral diseases. So it's remarkably easier for that than we would see for other. Train of thought there for a moment. So I'm just going to kick off. The other thing I want to make note of and mention of is a lot of the information I'm going to provide to you today is not always publicly shared in a rapid period of time. So we depend right now on press releases from companies, from pharmaceutical companies, from researchers at an unprecedented amount. Normally, in preparing a presentation like this, I would go to peer-reviewed literature and provide you the most up-to-date information. Unfortunately, a lot of that information hasn't been disseminated just because of the rapid pace in these developments. And as a consequence, we are getting our information largely from the lay press, and that makes things very difficult in really discerning the practicality of certain treatments, the toxicity, side effects, and the efficacy of treatments and vaccines. And I think one of the things that I've noted since the start of this epidemic is what a difficulty bioarchives really presents in a circumstance where we have an emergency situation. There's been way too much uptake by the lay press in articles that are presented in bioarchives that are not peer-reviewed, and then as a consequence disseminates almost false information. And that is really problematic in the population really understanding what is going on in this pandemic. So for lack of a better place to get my information, I'm always looking at the New York Times for the most recent update. And I really encourage you to go to Corona Vaccine Tracker in the New York Times, because I have not found a more effective summary of what vaccines are in development. And this is just a short list. It's updated just about every day in regards to which vaccines are in phase 1, 2, or 3, and then also which ones are already in limited use. As we all know, the limited use vaccines, there is one in China in the CanSino vaccine, which is an adenovirus-based vaccine that is being in military personnel in China. And then there is also the Sputnik vaccine from Russia, which is also an adenovirus vaccine, which I'll kind of cover briefly just to give you an outline of that. So there's also and preclinical development. And that's interesting to note because we really sincerely hope, myself included, that every vaccine that's in development right now and in a phase 3 clinical trial will be effective. And I have a lot of confidence that the vaccines for human use will start rolling out in probably January to February. And the biggest obstacle we face is production. And as a consequence, that's why I'm much more in favor of the RNA-based vaccines, because I think they'll be much easier to produce and can be provided in a much more rapid pace to the population. There are aspects in relation to cold chain, which really limits their use in places like sub-Saharan Africa. So on this list in the New York Times, even we are included. We're developing a vaccine which is based on the vesicular stomatitis virus. And we are in the process of we made the vaccine constructs with the coat protein, which surrounds the coronavirus, the S protein, and then just the parts of the coronavirus proteins on the surface that might be most immunogenic and most protective in activities in the virus replicates. These have been constructed. They've passed through animal models now, and we're just completing the tests in the correlates of immune protection potentially, and also determining toxicity testing in small animals. And we should be entering into phase 1 clinical trials, hopefully by the end of the year. Why are we approaching a VSV-based vaccine? We work a lot with the developers in Canada of the VSV-based Ebola vaccine, which has been through phase 4 clinical trials and is used quite effectively in the population already. It's licensed to Merck. And this vaccine has an advantage because the VSV, this vesicular stomatitis virus, is non-pathogenic to humans, and also has a tendency to grow and to replicate in an immunized individual. So it provides a very large dose of the factors, the antigens that provide protection against, for example, the coronavirus. So it has been effective for Ebola, and it is used to help contain pandemics currently. I also work a lot with a colleague at Imperial College in London. I used to be a visiting professor there, and I don't help him at all with his RNA-based vaccine. But you might have heard of Robin Shattuck, for those who have been to the UK or work a lot with Warwick. And Robin is pursuing an RNA-based vaccine. I'm a good friend of Robin, so I'll put that as a conflict of interest. And I'm very hopeful for his vaccine, although he's slightly behind. He's in phase 1/2 clinical trials right now. But his vaccine, as opposed to the vaccines developed by Moderna and by Pfizer, which are also RNA-based vaccines, is a little unique in that it replicates. So similar to the VSV-based vaccine that I mentioned earlier, which can expand in an immunized individual, this self-amplifying RNA vaccine also expands mostly within the cells of the vaccinated individual, but it produces a greater antigen load, which should enhance immunity. And as I'll indicate in a couple of slides, these new technologies that are brought to bear on the coronavirus epidemic are really going to help vaccine development against all viruses in the future, I hope, and even parasitic infections. So there is a lot of good things happening based on this coronavirus epidemic, as opposed to the horrific mortality and infection and severe disease that we're seeing. So like Robin Shattuck's approach, which he has actually, I'll just go back a slide, he's partnered with a company called Morningside. And what they're doing is a non-for-profit distribution network. So especially to try to get this vaccine into the low to middle income countries in the future. And this is where I find the biggest problems we're going to face. As you know, most of the high-income countries have already secured their dosing from the multiple companies that are in phase 3 clinical trials. And this creates a huge dilemma for the low to middle income countries like in sub-Saharan Africa in how we will distribute this and how will we control their pandemic. As you know, this is a global pandemic, as Saverio described. And you do not control a pandemic by just vaccinating your own population. It's too small and travel is too extensive. Even our attempts in limit foreign travel has been ineffective, and most of our cases are coming from foreign visitors. And as a consequence, we need to be able to eradicate this virus across the globe, and we cannot focus just on the high-income countries. So Pfizer, I won't go through this in any detail. Pfizer is also approaching the RNA vaccine field with an mRNA vaccine similar to Moderna. It is moved into phase 2-3 clinical trials. There are up to 30,000 participants in this trial, and I think you're going to see the results emerging from these trials by the end of the year, not just Pfizer, but also for Moderna. And this is one caveat that we, again, have to pay close attention to in the development of these vaccines. There is an incredible amount of financial pressure on these companies to report strong and to move their product forward. These are, even in the short period of time in this development process, these are close to $1 billion of investment that companies are making in getting these vaccines out. So we have so much competition in developing vaccines that I sometimes fear that certain data may be misleading in this approach. Now, the pharma companies, the major ones, have signed agreements now to be very forthcoming in their data and to ensure safety and efficacy in any vaccine that moves forward. I hope that's going to happen. So as many of you know, Moderna is the one that often gets a lot of attention. Moderna received at least $1 billion of investment from the US government in its development. And I think these are good things in the sense that we really need to push these vaccines, but we also need very strong adjudication of safety and efficacy, and that's where some of the issues now lie. But I do see trends that are very promising, especially with the AstraZeneca vaccine in halting that study when you have an adverse event. And just to put some favor on the US FDA still hasn't reinitiated that phase 3 clinical trial. Moderna has a little bit of an issue in their development, and I'm a little concerned because they recently lost a patent dispute, and that patent dispute was based on their delivery mechanism of their mRNA vaccine. And as a consequence, Moderna is now concerned because a lot of their revenue over the potential vaccine may actually go to a second party that developed their delivery system. So this is something that's working through the courts in the US. I'm most frustrated by this because now we're talking about financial and market pressures on Moderna, and they may have the most effective vaccine that can be delivered the easiest and doses produced by the simplest mechanisms, as opposed to a lot of the viral-based vaccines where millions and hundreds of millions of doses will take much more effort to produce. So time will tell how this will all shape up in terms of the effectiveness, and also if this will come to fruition. Very brief slide on how these RNA vaccines work, because you hear about it, and if you've taken simple biology, I can explain it to you in a matter of seconds. So basically, you have an mRNA, And that mRNA is encapsulated in a lipid bilayer, so much similar to what cells are encapsulated in. This allows for a very effective delivery system. So this mRNA encapsulated in this lipid bilayer is relatively stable for immunization. Now, this little particle with the RNA gets taken up by cells following immunization, and it gets uncoated, and then the RNA, relatively naked, gets exposed in the cell. And then as a consequence, ribosomes that make our proteins translate that RNA to make viral proteins that mRNA is encoding. Those viral proteins from the coronavirus then have many different pathways for protection or to induce protection in our immune response. They can be presented on the cell surface for those who are familiar with MHC class 1, and as a consequence, induce a cell-mediated response. They can also be secreted, and if they're secreted like the S protein of coronavirus that sits on the surface, then you can get a strong antibody response if the right parts of the virus are presented to the immune system. So that's typically the way Pfizer and Moderna's vaccine works. The difference in the is this approach. You put an mRNA into a lipid capsule, and then it's delivered. The difference here is that you encode in that mRNA a protein that then amplifies that message. So it makes many copies of RNA in the cell. And then as a consequence, you can produce more protein, and it's presented in the same way that I mentioned above. So these are new vaccines. They've never been tried before in human diseases. And it's remarkable, I think, as how effective they are. And it could change the entire landscape of the way we develop vaccines in the future. And so again, it's amazing what coronavirus and SARS-CoV-2 has done to the biomedical industry in rapidly developing these types of vaccines. Then the last I'll present is vaccines. From AstraZeneca and University of Oxford. They use a chimpanzee adenovirus as their vector. They got huge investments from the UK and the US government in the development of those vaccines. And they also have preparations to produce millions and millions of doses with the assistance of Indian vaccine production firms. But as you know, there had been an adverse event in the UK just recently, which led to neurological consequences. This is always something that's very difficult to understand. We are in a blinded trial, and as a public, we have no idea if those neurological consequences are related to the actual vaccine or if they were in the placebo arm. So until that is been, that will have been determined from the people that are engaged in the study and that review the adverse event reactions. So it's good news that the trial has started up again in the UK. I'm a little concerned that the trial has not been reinitiated in the US. So there might be something that the FDA is seeing that they want to do more analyses on why there was this potential complication. So we have to watch for that in the future. Johnson & Johnson, one of the biggest manufacturers of vaccines, along with Merck, is also working on an Ad26 vaccine. This is similar to what the Russians are doing. They just launched their phase 3 clinical trial, there is a lot of similarities between the Sputnik vaccine, if you will, the vaccine from Johnson & Johnson, and the vaccine from AstraZeneca. We'll have to see what the results are. Now, there's been a lot of debate about Sputnik 5 and the safety and efficacy of that vaccine. They've recently published an article in Lancet assessing the safety in their phase 1-2 studies. I've looked at it, unfortunately with phase 1-2 studies, which are safety-based, there's very little information, there's very little information provided on efficacy, mostly just on safety. So it's really hard to assess whether this vaccine will be effective. There is a part of this approach that I really do like, and that is by priming and then boosting with the vaccine in regards to using a different vector. And that is really, sorry, just leaving a note. That is really a good approach, because when you prime and boost with a vaccine, that is a different vector than with, it's a little complicated, but you develop an immune response as well to the proteins from the coronavirus that's produced by this vector, but you also develop an immune response to the vector. So when you boost with the same vector that's producing the same protein the second time, you often get an immune response to the first vector, which is the same. So you actually diminish your immune response because of an immune response to that same vector. So that's complicated. I'm sorry, I got lost in that description, but there's a real advantage of using two different vectors in the prime boost strategy that is basically doing the same thing in response with something to recognize from the coronavirus. We're using a similar approach with VSV by using two strains of vesicular stomatitis virus for presentation. So I, unlike a lot of people, have some faith in what the Russian government is involved in here. Now, of course, a lot of their safety data, I think, because so much of it was out there, may have been stolen from various companies and researchers around the world to determine if their approach would be safe. I have no proof of that, but it seems like that is quite possible based on news reporting. So I am going to where am I? Okay, so the one thing I would say about the adenovirus-based vaccines is that there is a lot of evidence right now that they provide some protective immune response, but they haven't fully protected animals from challenge virus. So there is some concerns related to that. Okay, so just a couple last slides in understanding where we are with drugs currently to SARS-CoV-2. And I have to say that unlike the vaccine approaches, drugs are much harder sometimes to develop. And as a consequence, there's much less information provided on there. Right now, we have very few drugs that are in the pipeline that have a strong promise. One of the ones that were recently published was a Merck compound that is a ribonucleoside. It's a small molecule inhibitor, and that showed relatively good efficiency of inhibition with minimal toxicity. And this was published now back in the early summer, and very little has been relayed about this potential drug currently. So I'm a bit concerned as to where it stands in clinical development. Ivermectin, ivermectin is a drug. It's an antiparasitic drug. It has shown some promise. Aside from hydroxychloroquine, which showed similar promise in sort of laboratory-based studies, but as we know, because of cardiovascular complications, was less effective. And I won't go through the mechanism, but surprisingly, it works quite effectively. But again, these studies were published very early on, have not been necessarily replicated in the primary literature, and it remains a concern as to their effectiveness. And then the one we know the best is remdesivir. It's a drug by Gilead. It was originally developed for Hep C and Ebola. It was not very effective in those diseases and was then brought in to utilize for inhibition of SARS-CoV-2. This is just a very simple diagram. This is our results using remdesivir. I didn't provide all the details of these studies. This is a very simple experiment as a control. And I can tell you that we've replicated the data that appears in literature, but it is not a very potent drug, quite clearly. And as a consequence, the effectiveness in patients is very low and just staves off the severity of disease. For a company, we are testing other drugs, numerous other drugs, and we can find compounds that are extremely effective and that have high delivery systems and that have very low toxicity. But I have to caution you that although the effectiveness of these drugs are incredible, it still takes a very long time to bring it through the pipeline of development, especially if those drugs are relatively novel in their activities. So I'm not going to go through this in any detail. I'm going to skip through these slides. The one thing I was just going to mention very briefly, like Saverio indicated about Western, we are somewhat uniquely situated just prior to the pandemic, and not with any forethought that a pandemic was emerging. We developed a state-of-the-art facility that is only replicated basically one other place in the world. We have a facility that's about 10,000 square feet that took about $8 or $9 million to construct, and we put in $12 million worth of imaging equipment in this facility. And we are fully capable of animal-based studies and tissue culture, and a vector production facility for GLP-grade vector production. So what this allows us to do is do pathogen-related research with the most modern technology that has often been our field of research. This opening this facility just prior to the pandemic is one of the craziest things I think we could have done. And as a consequence, the amount of work that we're doing in relation to prevention, in relation to providing prevention, drug testing, vaccine development, and just regular pathogenesis studies is astronomical. In just a few months, we obtained about $3 million in grants and contracts. We work with about 15 different companies, and it's just so rapid. I just can't keep track of everything anymore. It's a very nice condition to be in some ways, but it's also very, so I'll just, these are just the imaging equipment. This is from another presentation, so I apologize. This is our level 3 facility. We have great capabilities of producing virus, monitoring virus. We have a fluorescently tagged virus that we can analyze it in animals. And one of my pet projects, which I will just finish off by saying, is doing these studies with a set of engineers to really assess the aspects related to aerosolization and transmission. So we've constructed aerosolization chambers, and I'm working with engineers that are fluid dynamic experts, and also that work on cough dynamics and transmission. A paper was recently published on understanding the dynamics of these coughs. We're using these them with virus, spraying it, and then through repeating or mimicking respiration, human respiration, through masks and through different materials, looking at how the virus is transmitted and what level of protection these different materials provide. In addition, we change the temperature, humidity, airflow conditions, pressures, etc., to really get an assessment of how transmission actually occurs and what provides the best protection. So I'm going to leave it there. I've talked enough. And sorry, I'm at my cottage in Northern Ontario. So I'll just show you a nice picture if you can see that. I don't know, but you can see the lake. And that's to do my Zoom calls with in the background. So thank you for your attention, and I'll leave it to the questions.
Okay, thanks so much, Saverio and Eric. They were excellent, outstanding presentations. And yeah, I'd like to put the background of your lake and landscape there to the back of my WebEx view, I think. So we've had a few questions. I wanted to start first to both of you, actually. What do you think it is that makes the elderly so vulnerable to the virus? For example, is it the decline in respiratory function with age that predisposes to the pathogenesis, or what do you think?
I'll let Saverio handle that one. So, Saverio, you're muted.
There we go. There was also. Can you hear me?
Yeah, we can hear you.
Yeah, there was also a question from the participants, and thanks all of you for attending this seminar. I think there was a question about whether the age the demographics of the Western countries was the major driver of higher mortality rates in the West as compared to even Qatar. And that's definitely one important explanation. Median age in Qatar, I think, is just over 33. In Canada is 41. In Italy is 46. So obviously, the demographics and structure of the population has played a very important role in driving different mortality rates across country. In terms of why elderly are more vulnerable, I mean, and this applies to some degree also to influenza and other viral infection, respiratory viral infections, which is the reason also why there is a widespread recommendation of flu vaccination in the elderly or people with high prevalence of chronic comorbidity. So that's the other major thing that we have observed across different countries, especially in the West, that the multimorbidity, so the presence of concurrent chronic disease, multiple chronic disease, is a major risk factor for increase, adverse effects from COVID-19, up to increased risk of mortality. I think I was mentioning to Naila, the increased risk of mortality goes up above the age of 70. That's what we have consistently observed in many countries, including China, and obviously Western Europe. In terms of the mechanism, Paul, I think the respiratory capacity of elderly may be compromised in terms of response to this viral infection. I will not comment on immunological mechanisms or things like that, because it's not really my field. But from an epidemiological perspective, I think there is a strong correlation with the presence of chronic comorbidities. I mean, that's and even the fatalities we have observed among people in the 40s or 50s, which occurred as well. I mean, most of them were closely linked to the presence of chronic comorbidities.
And can I ask a follow-up question? Of course, in the Middle East, we don't have care homes for the elderly. The elderly are cared in the extended family. And do you think this type of care provision for the care for the elderly with their close extended family relatives may be identifying decline in health earlier than when people are together in care homes? And of course, in care homes, they're together, close proximity, and it's been a ripe for spreading transmission of the virus. Do you think this different approach of care for the elderly has helped keep the infectivity down in the Middle East and Eastern countries?
This is, I think, this can work both ways, because to some degree also in some parts of Southern Europe, including Italy, there is a high presence of intergeneration families. And that's been one potential explanation, for example, in Italy, about the high, mortality rates, again, especially among the elderly, because there is more interactions, within families between grandchildren and grandparents. So I guess this may work both ways, because obviously, again, in Italy or in some of the Western countries, that has been one of the potential explanation for high mortality rates. But on the other end, I mean, you made the point that elderly living in long-term care facilities, especially if they share, for example, the same bedroom, which unfortunately does happen even in Western countries, and also there is a interaction with workers coming from the outside community, and the lack of PPE, the lack of qualified personnel, the underfunded structure of long-term care facility, which is a problem, as you know, in England, in Western Europe, and in Canada as well. I think all these factors have played a role in inflating mortality rates within those settings. Whether or not being in family as opposed to long is a protective factor, I think is very context specific. So I will not make general comments.
Yeah, I would just add to that in the sense that you look at a lot of the countries in the Middle East, they were very quick to close their schools, and consequence that might have resulted in reduced transmissions in the familial units. I think in relation to the first question, not severe disease, especially with related comorbidities, but why don't infants? And that is usually the trend with most respiratory diseases like RSV and influenza. And I think it's twofold. One is the underlying conditions, but it's also if you want to talk about mechanism, it is the aspect related to immune inflammation. And because children tend to have a reduced antibody response and reduced immune inflammation, they succumb less to disease. And in the adults, that's why I think dexamethasone and other anti-inflammatories have been somewhat effective in reducing the severity of disease. It's also one of the reasons why the mortality rates have decreased as this pandemic has gone along. And then finally, look at the patient demographics in India and other countries where there's not as high of an elderly population. And unfortunately, because severe mortality related to other comorbidities and other diseases often reduce that population that might be affected, really results in a relatively low mortality rate in low to middle income countries, which is kind of unexpected, if you will.
We had a more general question really about vaccines, wanting to know how they, in general, do they protect a person against COVID-19 disease and pathogenesis?
Yeah, so almost all of them, basically the ones we think will be most effective is the ones that develop a strong antibody response that also produce what we call neutralizing antibodies. So antibodies that are produced that when they bind to the virus can prevent them from infecting cells and effectively kills the virus, if you will, because it can't replicate. There are other immune mechanisms that are at play that are often related to antibodies that might provide protection. I think one of the biggest fears we have, and that's one of the reasons why we're very careful in determining efficacy, is the potential for detrimental antibodies like we see with other diseases like dengue and inability to produce vaccines against them. So we want to make sure that we don't have a consequence with coronavirus. So far, so good. I think, I always argue that if we had done more with this first SARS epidemic in developing a vaccine and really continued in that development effort, we would not be in the same circumstance. And fortunately, we did do a little bit of work on SARS and MERS and vaccine development that really pushed us ahead with this particular epidemic. And I really hope that in the future, you know, we will be better prepared and we invest them now instead of trying to play catch up all the time.
And we also have a question about the situation, as you understand it, in the MENA region, Qatar and the MENA region. I think it's one of the Sinopharm vaccines is being tested in UAE, I think they say. I saw in a review in Nature Immunology. Do you know anything about the vaccine trials affecting the MENA region?
I'm sorry, I don't always remember all of the details, but I know a couple of the Chinese vaccines are being tested in the Middle East, and even the advirus vaccine from Russia is being tested in some countries like Mexico, I think in some Middle Eastern countries, but I can't remember the details. All being tested in trials. You know, I think it's unfortunate, but I don't always the development and testing of these vaccines is always based on rational scientific approach, but often due to political pressures as to who and where vaccines get tested. And if I look at where the Russian-based vaccine is being tested right now, it's often in countries that may be more influenced by the Russian sphere of influence than other vaccines. Now, I like the Russian vaccine approach, and if their safety is true and effective, I think it might do quite well. I think we have to be, you know, cognizant of that, but they definitely took shortcuts. And the Chinese vaccines, unfortunately, similar in the situation, really trying to push the envelope a bit too fast.
Yeah, and you mentioned about the drug evaluation. I know we're doing some studies actually in collaboration with Qatar University, where we have a high containment facility in the virology laboratories there. And we use these viro cells that you mentioned, but they lack the proteases that provide for one fusion pathway. They only have the endocytic pathway. And I think this is where the hydroxychloroquine was tested initially. You know, so if you only have the endocytic pathway, then the drugs blocking the endocytic pathway, of course, seem very potent. But in vivo, of course, you have this protease fusion pathway with the transmembrane serine protease and furin protease that provide another way of infection of host cells. So what do you think about that?
Yeah, so we, I didn't get into details. In our approach, we start off with the Viro E6 cells, and then we move to the Calu3 cells. Really get at the question that you're asking, especially when it's related to entry inhibitors and mechanisms related to entry. Yeah. I think with the drugs that are ribonucleoside analogs and some of the other ones that unfortunately I can't divulge because of CD8, but those ones tend to inhibit intercellularly and where that furin protease cleavage might be less of an issue. But I think it's very important that you test it in Keiko and Calu3 cells, and we follow Syrian hamster-based studies to really assess effects. But, you know, I think you probably realize this as well as I do, is the development, especially of these novel inhibitors, is going to be so difficult to deal with this current pandemic because the timelines are so extended compared to vaccines. And you can even see the companies just is primarily on the vaccines rather than on the drug development. Unless you can pull something off the shelf that's already in human use, there's not a lot. So remdesivir was, you know, a reasonable approach, but I would say it's less than impressive in terms of its inhibitory potential.
Yeah. We have another immunological related question. Are there targeted delivery strategies for COVID-19 mRNA vaccines to specific antigen presenting cells for increased efficacy?
So maybe I unfortunately was too remedial in the presentation, because it's a great question. So far, I think it's where you do the immunization, epidermal, subdermal, intramuscular immunizations to get the best antigen presentation. Now, there is a possibility to put proteins onto the liposomes that carry mRNA-based vaccines to allow for potential targeting into antigen-presenting cells. I haven't seen anything like that in this particular round of vaccines. I think we're looking at sort of the bare bones of the mRNA vaccines, because it's such a, well, it's a relatively novel technology to use for human trials, let's say. And I would say that if it wasn't for coronavirus, I don't know how long it would have been before the mRNA vaccines would have been adopted. And I really am excited by that, because especially for production, it's so much easier than producing a vaccine that's viral.
We yesterday.
There.
Yeah.
Sorry.
Okay. Yeah. Only yesterday we got a paper accepted that provides some background for repurposing anti-cancer drugs for treatment of SARS-CoV-2, modifying SARS-CoV-2 and treatment for COVID-19. So we're continuing some studies with Qatar University on that. Some reactive metabolites that are increased in response to some anti-cancer drugs will modify the SARS-CoV-2 proteins, particularly the spike protein and the nuclear protein. And one other area we've been looking at is I'm interested, particularly myself, in the age-dependent decline in lung function, where interleukin 8 and MCP1 are thought to be major mediators, bringing macrophages and neutrophils into the lung, producing fibrosis and inflammation. And we have a dietary supplement that decreases the levels of these inflammatory mediators, and so maybe used as a supplement, dietary supplement for the elderly, to help maintain their lung function. So what do you think?
I'd be very interested to see your recent study. I think repurposing drugs, chemotherapeutics, various drugs to me is the approach to use in the short term. Absolutely. And to reduce inflammatory response seems to be the best that we can provide. And reducing chemotactic responses that allow for infiltration in the lungs that cause that inflammation response is, quite interesting. And I always liken it to pregnant women that have no disease whatsoever. And if you look, they're more skewed to a Th2 type of response than a Th1 type of response. And, their inflammations are different. So the question is do we get information from that kind of understanding? Because to date, there's very few cases of pregnant women having severe disease. And it's just the opposite with most viral infections.
Okay, I think we can leave it there, just to say that we're all hoping very much and praying that we counter this pandemic well in time for the 2022 World Cup. And so we can welcome people from all over the world to our lovely stadiums and have a great time in the World Cup 2022. Hope we get there, and I hope you can come along. So thank you so much.
We will be there.
Yeah, Mario and I will come and visit, because I spend half of my research life in Uganda, and so I haven't done it yet, but one of the ways is to go through Qatar. So hopefully we'll visit.
Great. So thanks so much.